Welcome to Chapter 7. This is all about microbial genetics. We're going to be talking about DNA, RNA, protein synthesis. We'll talk about mutations. We'll talk about how we identify mutations and mutants and how we can use these things in biotechnology. So the first thing we like to always start out with is, why are we looking at this, right? What's the purpose of looking at these genetics? Well, bacteria and archaea have been around for billions of years. Eukaryotes have been around for a very short time compared to bacteria and archaeans. And humans, I mean, super short, right? So what makes them so able to survive? You know, the environment has changed so much. Our air, the ground that we are on, the temperature, everything changes all the time, but these organisms can just persist. Well, the answer comes from Charles Darwin, and one of his theories was the survival and reproduction of the fittest. So what this means is organisms that are best adapted to their environment can survive and then reproduce and produce offspring. So if you are adapted to survive in an environment, you'll probably survive. You have to be able to be healthy enough to also reproduce and pass on those genes. It's the genes that are living. The organisms, will pass on their genes to offspring and pass on their genes to offspring. But the genes just stay forever and they can adapt. So what are genes? Well, this came from Gregor Mendel. Gregor Mendel is father of genetics. He figured this out before we really knew a lot about what DNA was. We didn't know what was the characteristic that was being transferred from one organism to another, from parent to offspring. But he came up with this in the 1800s. He used pea plants, and he would take pea plants with different characteristics and cross them. And he would get unique characteristics or characteristics of either the mom or the dad plant. And so this is how he came up with the idea that these traits or these characteristics, which we now know are genes, are passed on from parent to offspring. So now we know that the genes are being passed on, we know that these characteristics are being passed on. We're not sure if it's genes yet. We just know that we have certain characteristics. And in the 40s, George Beadle and Edward Tatum, they started looking at genes, and they figured out that genes, produce proteins. They code for proteins. And what they found out was that specific genes code for specific proteins. So now we know that the DNA that carries the genetic code for all of our proteins, right, codes for all the proteins that we have, is an actual code that codes for specific proteins. So this section of our DNA, section 1 of chromosome 1 of our DNA codes for one certain type of protein. And then section 2 of chromosome 1 codes for another protein. And they were awarded the Nobel Prize in 1958 for their work. So we know that our DNA carries these genes. We know that the genes are coded for, we know that DNA carries our genes, and our genes are what code for proteins. So now we were trying to figure out, well, what does DNA look like? We don't really know a lot about DNA. And so everybody was rushing to try and figure out the actual structure of DNA, because whoever found it was going to be like famous. And in one laboratory, Rosalind Franklin and Maurice Wilkins worked together. In another laboratory, James Watson and Francis Crick worked together. And typically in laboratories, you didn't cross information. They didn't like to share information because they were all trying to figure out the structure on their own. So Rosalind was working with microarrays. She was looking at using X-ray diffraction to make a picture of DNA. While Watson and Crick knew or had figured out that DNA was composed of certain characters, and they were trying to figure out how do they put them together. Well, Wilkinson stole, he was working with Franklin, so I mean, he wasn't like stealing, like actual, he had papers that him and Rosalind were working on, and he shared that information with Watson and Crick. Now, Rosalind Franklin, she ended up dying when she was in her early 30s or mid 30s from cancer. And with that information that Wilkins gave, Watson and Crick were able to figure out the structure of DNA. And because of that, Watson, Crick, and Wilkins all received the Nobel Prize for discovering the DNA structure, which is a double helical structure. It looks a lot like this structure here. Obviously those are beads, but we know that adenine, thymine, guanine, and cytosine are what make up the structure, and I said these in a different way than they're actually written, I realize, sorry. I do want to mention that though, I'm going to go back to this just so I can talk about this again for a second. Though Rosalind had done the work and actually made the pictures, came up with how to identify the structure using X-ray diffraction, she was not able to receive the Peace Prize because she died, and you can't give a Nobel Prize to someone who's dead. But we like to remember her because she definitely played a part in identifying the structure. So now we know that DNA is a double helical structure with a backbone composed of carbohydrates and phosphates. So deoxyribose is the sugar, and the phosphates are phosphates. There are only 4 nucleotides, which you see there. Adenine and thymine always pair up. Guanine and cytosine always pair up. And because of this, because there are only 4 nucleotides, we have to read DNA 3 nucleotides at a time to code for all the different amino acids that we have, which we have a total of 20 amino acids. So bacteria and archaea have their DNA organized within a region of the cytoplasm called the nucleoid region. This region is not boxed off. There's nothing surrounding that region. It's still just part of the cytoplasm, but it's just where the bacterial DNA typically is found. Bacterial DNA is typically haploid, or it's a single chromosome, so haploid, and it's a circular chromosome. But bacteria and archaea can also contain extrachromosomal structures called plasmids. These are small circular structures that do carry sometimes important information and useful information, but not essential for the bacteria to survive. And so that's what our bacterial DNA looks like. DNA replication, you probably remember this from your Gen Bio class, or if you're in anatomy and physiology, you probably remember it from A&P class. So there are three basic processes for replication to occur, and this occurs both in eukaryotes and in our bacteria as well as archaea. The three processes are initiation, where we're going to unwind and unzip our DNA so that the DNA polymerase can bind. Elongation occurs as DNA polymerase moves along the chromosome, allowing new nucleotides to come along. And then the termination, when the DNA polymerase hits the end and comes off, and the new nucleotide, or I'm sorry, not nucleotides, the new DNA strands can fold up with their parent strand. And so here's what DNA looks like when it is in the nucleoid region. It is double-stranded, and it is in a helix, forming a helix. So we have to unwind and unzip it, and so this is what we're looking at here. DNA, I love this picture because it shows DNA it runs anti-parallel. So here's the 5' end down here of the blue strand, 3' end, 5' end, 3' end. So everything is opposite for each strand. And this process is called semi-conservative because one parent strand binds with a new daughter strand, producing two new DNA strands, double-stranded DNA molecules that consist each of a parent and a daughter strand. So what actually happens? We have to unbind the DNA and unzip the DNA. So DNA helicase comes along and unwinds the DNA. This and helps to unzip the DNA as well. This allows other molecules to come in. So topoisomerase breaks the DNA molecules so that the strands can separate. So now we have individual DNA strands. So either in that one picture you saw blue and red, the blue and the red would be completely separated now. We have an RNA primer that comes along and binds to the 3' end and produces a tiny section of DNA. Okay, so primase is an enzyme that produces this RNA primer. DNA polymerase can only bind to a double-stranded molecule. So the RNA primer produces that double-stranded molecule so DNA polymerase can bind. This occurs on the 3' end only. And then DNA polymerase comes along and attaches to the area where the RNA primer was. It digests the primer and then allows nucleotides to come in that are complementary to the parent strand that it's attached to. And so we end up having replication occur on both strands of the DNA, and the end result will be two identical circular strands of DNA that are double-stranded and form a helix. So if you look at, so you see this leading and lagging strand. I'm going to go back here. I want you to see this because I think this is an excellent picture. So you see where you have this DNA polymerase, RNA primer, and then on this side we have just DNA polymerase. We're only seeing one. DNA polymerase will bind to the 3' end only and produce new DNA strands in a 5' to 3' direction. The RNA primer will also only bind to the 3' end of the DNA. So when we unbind our DNA, one of those strands is going to be 5 to 3, and one of those strands is going to be 3 to 5. And so the RNA primer is going to be produced on the 3' end of each side. Whichever side the RNA primer binds that is open completely will be considered the leading strand because you'll produce an RNA primer, bind it, and then you'll produce a new parent strand, or a new daughter strand that is complementary continuously. So leading or continuous strand. Whereas the other strand, the lagging strand, each time we open the DNA a little bit more, a new primer can bind or can be produced, and then DNA polymerase has to bind, digest the RNA primer, and then produce the new DNA molecule. So that's called the lagging strand. And so here you can see it again, the leading strand and the lagging strand. The leading strand is continuously synthesized, whereas the lagging strand is synthesized discontinuously or in small sections. And then DNA ligase comes along and seals all the breaks. So replication allows the DNA to produce 2 copies, and then the cells can replicate. So for E. coli, chromosomal replication, it takes approximately 40 minutes, but the generation time of E. coli is 20 minutes, and that seems very unusual, because how can the bacteria replicate faster than the chromosomes replicate? The chromosomes have to be able to move into each cell, so that we have 2 identical daughter cells. Well, the answer comes in how replication occurs. As DNA is being replicated, before the initial round of replication of the DNA ends, new replication starts on the new cell. So replication is going that fast. So we're replicating the DNA to produce 2 identical copies, and then while those are replicating, we're replicating the daughter strands that are partially produced. And so this allows us to produce daughter E. coli cells that have partial replication already. So now we're going to get into protein synthesis. The central dogma of biology is DNA produces DNA produces or codes for RNA, which codes for proteins. So DNA carries the information of all of our genes. The information in our genes can't be produced by our genes directly. So it has to be transcribed into a new language called RNA, and then RNA can be translated into a protein. And this is protein synthesis. So transcription is the first step of protein synthesis. RNA polymerase binds to a DNA molecule, a single-stranded DNA molecule, so it opens up. It binds to the template strand and synthesizes a single strand of RNA. The direction that RNA polymerase runs is 5' to 3' direction, so it can only attach to a 3' end. It binds to a region on the DNA called the promoter, and it stops the transcription at a region on the DNA that's called the terminator. So we transcribe using DNA as our template. DNA is double-stranded, so it has to unwind so that RNA polymerase can bind. There are 2 strands then of the DNA. One of those strands is called the coding strand. The other strand is called the template strand. The coding strand is also known as the plus strand. The template strand is also known as the minus strand. The DNA strand is going to be identical to the messenger RNA strand if it is the coding strand. So the coding strand of DNA will be identical to the messenger RNA produced, except for anywhere that there is a thymine in DNA, we have a uracil in RNA. So we use the non-coding or the template strand to produce the messenger RNA molecule that is identical to the coding strand. And so if you look here, you see here's the plus strand, here's the minus strand, and here's the messenger RNA. Everywhere you see a G here, you see a G there. C, C, T, U, because we don't have thymines. G, G, A, T, U. Okay. So we use this strand, the minus strand, to make our RNA molecule. This allows complementary nucleotides to bind, and then we have this identical copy. Messenger RNA transcripts then are what we've produced, and they can be either monocistronic or polycistronic. Monocistronic messenger RNA is what we see typically with eukaryotic organisms. So humans produce monocistronic DNA or monocistronic messenger RNA, which means we're coding for only one gene. Every messenger RNA that we produce codes for one specific gene. Many bacteria are polycistronic, so they can produce messenger RNA molecules that code for multiple genes. So instead of just one gene on the messenger RNA, now we can code for 3 or 4 genes. This is important because a lot of times we need 3 or 4 genes to produce some function in the bacteria, and they have to work together. So for transcription to occur, again, we're going to have 3 basic steps. Initiation, where RNA polymerase binds to the promoter region. Elongation, where the RNA polymerase moves along the template strand and produces an identical copy, or not identical, a complementary copy of messenger RNA to that template strand. And then termination, when we hit the terminator sequence. So the promoter region on our chromosomes will orient the direction of transcription. So if the promoter region tells the messenger RNA which way it's going to run. So RNA can bind and then produces a single copy of some gene, and another RNA polymerase can bind and produce another copy of a different gene running in the opposite direction at the same exact time. So here we see the template strand, and then we see the coding strand. I've separated them. This is my own replication thing, okay? We have this region of DNA that doesn't really code for the protein. So we have these A's and C's, and then we start where we are actually coding for the protein. TAC is the first codon that we use, and it codes for AUG. AUG, if you look in a codon table, codes for methionine. All right. So this region here is called the promoter, and there's the leader strand. This is where RNA polymerase binds, okay? And then it's going to move along here and allow us to produce a complement to the template, which will be identical to the coding strand other than those T's and U's. Let's watch this video quickly.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of messenger RNA called the sigma participates in recognition of the promoter region. Soon after transcription is initiated, the sigma Synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is either byproduct or three-prime enrichment. Messenger RNA to dissociate from the DNA.
This table that you see doesn't actually come from your textbook, but I like it because it talks about a lot of the materials we'll talk about now. So we'll talk about anticodons and how they function in the process of translation. We'll talk about tRNA, so this is what it is. tRNA is a molecule that acts as a key, carries a specific amino acid, and it also has an anticodon, which you can see on here is on the tRNA. So it's just nice because it gives you a little bit of information about each of these regions or each of these components of translation and the importance of them, okay? So we've transcribed our DNA into messenger RNA. And when I think of transcription, I always think of transcribing from one language to another. So our DNA is this really important material that our cells can't use. It's like super, super awesome, but nothing can read it. Nothing can use it except for our RNA, our ribosomal, or our RNA polymerase. It can read it and produce a transcript that is in a language that the rest of our cells can actually read. And so a transcription is basically taking and copying a language that we don't, or that our cells don't understand, and producing a language that we do understand. So we have to do this all the time. Think about when your kid brings home math homework and you're looking at what he has to do and you're like, I have no clue. And then you have to figure out, transcribe the information so you understand it. Another even probably better example, especially for people who are like into acting and stuff, when you get a play and it's in one language and you have to transcribe it to another language so you can actually produce the play. I always use the transcription of the instruction manual for like building a crib, because when you have to build a crib, good Lord, the instruction manual is crazy. That's all I have to say about that. And so you have to take that and you have to try to decode it or try to transcribe it into something we understand. And so typically we use the pictures because everybody understands pictures. And if it has a picture of, a metal rod with the letter A, and we find a metal rod that has the letter A, we're like, okay, this is what they're talking about here. Oh, we have to put A into B. I got it. We can do that instead of trying to read the information and understand it. And so if transcription is the producing a language that is understandable by the cells, translation then is producing the actual proteins. So with translation, we have to use amino acids. And if you remember from the very beginning of this lecture, I had mentioned that the number of amino acids we have is 20. And we use a codon table that codes for each amino acid. So these codons come from our DNA or RNA, however you want. You can have a DNA codon table or an RNA codon table. Typically we use RNA codon tables, I guess. but the information comes from our messenger RNA molecules or the DNA. And so a codon that's called a sense codon is one that codes for an amino acid. There are 61 of these. Then we have 3 that are called nonsense codons. Nonsense codons do not code for amino acids, but they stop translation from occurring. There are 3 nonsense codons. So these are what terminate translation. Okay. And then the genetic code uses 3 nucleotides at a time to produce a codon. We figured this out because if we were only using 1 nucleotide at a time, we would only be able to produce 4 amino acids because we have A, T, G, C, right? So each of those codes for 1 amino acid. So we thought, okay, what if we use 2 nucleotides? So then it'd be 2 to the 4th power, right? 2 times 2 is 4, times 2 is 8, times 2 is 16. We have 20 amino acids, so 16 is not enough, which is why we use 3 nucleotides. This actually gives us 64 different codes, 61 code for amino acids, and 3 are stop codes. This makes the genetic code very redundant. And that's important because that means when we do have a mutation, in general, the mutation can be considered a silent mutation. So here's a codon table. Here we have the first column, here's the first row, and here's the last column. In the first column, we have the first letter of our DNA, or our RNA, UCAG. In the first row up here, we have the second letter, UCAG. And then in the last column, we have the third letter for each section. So UCAG, UCAG, blah, and blah, right? So when we're looking for any given amino acid, let's just pick 3 letters, AUA, okay, AUA. We have to look here, and we go down to where A is, A. And then we have to look up here to see where the U is. There's the U, so we have to go down here again. So we know it's in this group. And then we go over here, UCAG, so AUA is right there. And that's isoleucine. Now, this amino acid here is circled, and it has the word start next to it. Methionine, AUG, starts every protein. In humans, it's the start amino acid. In bacteria, it's also the start amino acid. So AUG starts all protein synthesis. And then over here, you can see UAA, UAG, and UGA are all stop codes. So they don't code for amino acids, they just stop translation from occurring. This leads us to the wobble hypothesis. The wobble hypothesis states that the last nucleotide is very loosely paired, and that allows different nucleotides to fit into that third spot and still produce the exact same amino acid. So the third position, not near as important, but the first and second positions are very important. And this eliminates our need for an individual transfer RNA for every single codon. We only need 20 different transfer RNAs that code for the different amino acids. Or that carry the anticodon and carry the 20 different amino acids. And so let's look back here really quick. Here's an example, isoleucine. Isoleucine has A and U in every single case, but the last letter, U, C, and A differ, but they all still code for isoleucine. Another example, threonine. ACU, ACC, and ACA code, and ACG codes for isoleucine, or threonine, I'm sorry. Some of them only have one code though. So an example, methionine. AUG is the only thing that codes for methionine. Here's glutamate. Glutamate only has two codons that code for it. And this is what helps to decrease mutations. So the nucleotide sequence determines the coding region, and we read our DNA 3 letters at a time. That is our reading frame, 3 letters at a time, the reading frame, okay? The sequence will designate the beginning and the end of the region being translated. So we have the start codon, and then we move all the way down to the stop. Okay. Oh, do I want to say anything else? I'm sorry. Nope, not on that one. I'm sorry. It's coming up. So ribosomes are what are the machines needed for translation. So they're basically the cellular machinery of protein synthesis. And you remember that from when we went over the different structures of both eukaryotes and prokaryotic cells, and we talked about ribosomes. All ribosomes work in protein synthesis. I'm drinking water and coffee, nothing bad. Of course. So, prokaryotic ribosomes are called 70S ribosomes, and they consist of a 50S subunit and a 30S subunit. These ribosomes will then attach to the messenger RNA that's produced, and they will produce they will allow for transfer RNAs to come in and bring the proper amino acids as they move along the messenger RNA. The ribosomes move in a 5' to 3' direction. So transfer RNAs bring in the proper amino acid being coded for in the messenger RNA, and they have an anticodon, or the complement to the codon, where they can actually bind properly. And this allows the proper transfer RNAs to bind in the proper region so that the correct amino acid is produced each time. So translation in prokaryotic organisms can actually start before transcription is complete. And so here what you see is RNA polymerase binding to the DNA, producing a messenger RNA copy, and then you see on here 1, 2, 3, 4, 5 different ribosomes moving along, producing the proteins in this messenger RNA molecule. So this, because there's so many different ribosomes, is termed a polyribosome, where you're producing multiple copies at the same time. So for translation, we still have initiation, elongation, and termination. Initiation starts when the ribosome comes together at the first sequence. And so I typically don't talk about the E site. I usually talk only about the P and the A site. So this is the P site, this is the A site. The P site is where the first amino acid attached to its transfer RNA comes in and binds using its anticodon. Then the A site gets filled up by its transfer RNA that carries the proper anticodon and its amino acid. Once both of these sites are filled, then a peptide bond will form between this amino acid called methionine and another amino acid. And in this case, it's proline. So once the bond is formed, then the entire messenger RNA, or I'm sorry, not messenger RNA, the entire ribosome shifts over one codon. So there's your peptide bond formed, and now we're going to have the ribosome move down one side. And so this first one that was at the initial P site is released and is able to go move to the amino acid pool and pick up a new amino acid. And so now we have the proline transfer RNA is now in the P site, and we have both the methionine and proline that are bounded together. Now here comes another transfer RNA, but moving to the A site with tyrosine. And so again, we're going to have a peptide bond form between the proline and the methionine to the tyrosine. And this will continue, and now here comes glutamate, and then this is going to continue until we hit a stop code. So UAG is one of these stop codes. What this means is once we hit the stop code, that's called termination, and the ribosome will come apart. The transfer RNAs are released to move back to their amino acid pool, and this polypeptide, multiple amino acids, can fold up into its functional form. And there you can see the ribosome coming apart, the amino acids bounded as a polypeptide chain. The messenger RNA can be retranslated if we want to. The transfer RNAs are going to move to the amino acid pool and pick up new amino acids. So this is a video. I'm not going to watch it here, but it's a really fun video that goes over transcription and translation. Okay? And so here we have translation. I drew this using different figures and stuff, so it looks kind of messy, but here's your 50S subunit, here's your 30S subunit for the ribosome. These are your transfer RNAs, okay, and then this is your messenger RNA. And so here we have methionine and glycine, and now we have asparagine sitting there. Phenylalanine comes along. Methionine, lysine. Hold on. I forgot asparagine there, didn't I? I didn't put him on there. There's leucine, lysine, blah, 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 all the way to the end until we hit that stop code, and we have a perfectly functional polypeptide that can be folded into its functional form. I'm going to stop here for the day, and you can watch this video, and then I'll set up the next video so that you can watch the next video in hopefully a few days, okay? Bye.